Epigenetics A SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding how phenotype is the result of an interaction between genotype and the environment. We'll also look at how epigenetic changes, including DNA methylation and histone modification, can modify the activation of certain genes. We'll also understand how epigenetic changes can be passed on following cell division, and also understand how phenotypes are affected by multiple genes for the same gene and many loci, which is polygenic inheritance, and how this can give rise to phenotypes that show continuous variation. So the epigenome are instructions that determine what genes and how they will be expressed in cells. So you can think of them like a user manual for DNA. And these epigenetic markers are copied and passed on through DNA replication. Different ways to control the transcription of DNA are DNA methylation and DNA acetylation. Another topic that so many students cannot wrap their head around, or it is very difficult to wrap their head around, so keep an eye out for this one. So epigenetic changes can be passed on through mitosis, as the result of mitosis are two identical daughter cells. Hence, epigenetic changes develop as the result of environmental changes before mitosis will be passed on to the daughter cells, preparing the daughter cells against changes in the environment. Some epigenetic changes are removed because after fertilization, the egg divides to produce totipotent stem cells, resulting in some epigenetic changes being removed. So, epigenetic changes do not alter the actual DNA-based sequence. This makes them different to mutations. So, the difference between epigenetic changes and mutations is that mutation actually changes the DNA-based sequence. So epigenetic changes deal with how a polypeptide is produced, while mutations change the actual polypeptide that is produced. So epigenetic changes just change the degree to which a gene is expressed. So you can think of it like the rate at which the gene is expressed. So yeah, like I previously mentioned, epigenetics is more like a user manual. It's like more of like a how-to guide, and mutations are more of like to do with what is produced. So it determines what genes are expressed, not the DNA-based sequence. So yeah. So what is DNA methylation? So this is the process of attaching a methyl group to a cytosine next to a guanine to prevent transcription, as it prevents the binding of the RNA polymerase enzyme to DNA. So if you guys notice from this video and the video on operons, which should be uploaded by now, is that the way to prevent transcription is literally by preventing RNA polymerase to bind to DNA. So when you talk about all these different processes like DNA methylation, DNA acetylation, or in the case of operons, what you need to think about is connected to the RNA polymerase binding to the DNA, or if it's RNA polymerase not being able to bind to DNA. So mention it somewhere in your answer when you talk about these processes. So anyway, back to DNA methylation. So this is because the attaching of the methyl group changes the shape of the cytosine, which changes the binding site of the DNA where the RNA polymerase binds. So DNA demethylation, so it's quite self-explanatory what it is from its name. So this is the process by which the methyl groups bonded to the DNA are removed. So RNA polymerase, again, with all these processes, what matters is the RNA polymerase binding to the DNA or is it not binding to the DNA? So just remember that one. Keep an eye out for that one. So RNA polymerase binds to the DNA, which switches on the genes associated to remove the methyl groups. So DNA acetylation. So gene expression can be controlled by activating or inactivating the genes. So this can be done by loose wrapping with the histone, which is a protein that provides structural support for a chromosome. It packages DNA by wounding tightly around the DNA. This loosening makes the chromatin less condensed and RNA polymerase is able to bind to the DNA. So now we're looking at the difference between phenotype and genotype. This isn't too different from what you're familiar with in GCSE, but we're going to go through it anyway. So genotype is basically an organism based on the alleles they contain. Phenotype can be described as the physical characteristics of an organism that arise as a result of genes that the organism possesses in combination with environmental factors. Basically, genotype deals with genes and phenotype deals with environment. So when characteristics are influenced both by genes and the environment, this variation is called continuous variation. And this is because there are multiple factors that influence the characteristic. And an example of continuous variation would be height. Your height is not determined by one gene, it is determined by a lot of different genes and a lot of different environmental factors as well. So that's what makes it a continuous variation. Also, you do not have specific categories of what height you can fall into. So there is variation in what are the different values of height you can have. You can be 5'4", you can be 5'11". It's not into groups like, for example, height A, height B, height C. So yeah, that's why it's continuous variation. So discontinuous or discrete variation deals with characteristics that fall into distinct categories. So the environmental influences on these are minimal. So for example, this would be eye color. So for discontinuous variation, you have distinct categories that it can fall into. So you can have brown eyes, you can have blue eyes, you can have green eyes. You cannot have like any other different categories of eye color. For example, you cannot be green 0.5 eye color or something like that. Doesn't make sense, but yeah, that's what makes it discrete or discontinuous. And another example of discrete variation would be your blood group. So the additive effect of genes. So the height of a plant is controlled by two unlinked genes, and these two genes have an additive effect. The recessive alleles contribute X centimeter, while the dominant allele, for example, contributes twice the amount. So this is a hypothetical example. It's not actually facts. So yeah, so basically recessive allele produces a certain amount of height, while the dominant allele produces possibly a larger amount of height. So different combinations of these can contribute to different heights. So which is why the height would be described as continuous variation. So if you want to actually look at what are the different heights you can have, then you can like give different values to the unlinked gene pairs and you can add them and see how it works. So monohybrid inheritance. So monohybrid inheritance is when the characteristics passed down from parent to offspring is influenced by one gene. So the clue here is mono. Mono means one, so yeah, it is influenced by one gene only. So each locus, which basically means position of a gene on a chromosome of the gene, is responsible for this feature. So what is polygenic inheritance then? So again, as the name suggests, it is characteristics that arise as a result of multiple genes at different loci, and this is what leads to continuous variation. Professor Snap today. Hope you found this video useful. If you have any questions, let me know in the comments below, and I'll get back to them as soon as possible. And don't forget to like, share, and subscribe.